You start recording. So yesterday you went over dihybrid crosses. You had an introduction to those. Today you're going to go. You have some practice. Do you see what's in front of you there? We're going to walk through some simple stuff, and then tomorrow we'll do an actual dihybrid cross activity. Okay. So to start, before we go to dihybrid cross, we're going to talk about something called a monohybrid. And in order to really think about that, let's take a look at this di versus a mono. What do you guys think? When you hear the word hybrid, first of all, hybrid, what do you get? Yeah, you would think mix, right? Hybrid means mix. And what about the prefix mono? One, something with one, a mixture of one. And what do you think a prefix di means? Two. So a mix of two. Okay? Is that making sense to everybody? So I'm gonna go ahead and write that down so we're clear on that. This means a mix of one. That seems weird, right? How can you mix one, right? And then here is a mix of two. Now that makes a little more sense because it's two. You can mix up two. To mix up one is really strange until you think about what we're talking about. So let's go back and review really quickly. We're talking about a chromosome that you inherited, one from mom and one from dad. We're talking about in mono, we're talking about one gene. So mono means one gene equals one, or equals mono, right? One gene. That's why we say mono. Why do we say mix? Why do we say hybrid? What are we hybridizing?
Are you combining the mom and the dad together?
Yeah, what are those things called? The two alleles. Excellent. So, but we do have two alleles, and we're mixing those, right? So we're mixing those two alleles. That's why we call it a monohybrid. We're only dealing with one gene. Another word that they like to use here is a locus, a location on the chromosome. Why is the gene called a location on the chromosome? Why are sometimes those synonymous? Because every gene is always in the same place at the same, on everyone's chromosome. So the locus is usually the gene. If the gene is there, we know it's there. It's going to be there. Now, are there times when they get moved around? Yes, but let's not talk about that, okay? So we usually call it a gene. So we can mix them up. Again, what are some of the possibilities? You can have big A or little a, right? There could be a bunch of different alleles, but usually they can be big A or little a, right? That's what you're going to see on your test. It's going to be a dominant recessive. It could be codominant. There's all kinds of options they can put on that test. But rarely are you going to see unless they do ABO blood groups like we've discussed, right? Because in ABO blood groups, there would be three, right? There'd be A, there'd be B, or there'd be little I, right? So in a dihybrid cross, though, you're still dealing with chromosomes, one from mom and one from dad. But what do you think's happening in a dihybrid cross?
They do it twice?
It's close.
They do both.
You do two what?
Two genes.
Two genes. And of course, those two genes have two or more alleles. two or more mutations, right? So again, we could deal with here would be either big A or little a, and the same here, right? Big A or little a. So that's one gene, but then you could also have a gene here because it's dihybrid, meaning we're going to concern ourselves with more than one gene. Here, this one could also have a big B or little b. All right? So now with a dihybrid cross, you're dealing with two genes. And there's trihybrid crosses. You know, there's a lot of genes on a chromosome. So there's a lot of different options you can get, a lot of different mixing up that can happen, especially if we get crossing over happening. All right. Before we go on, let's talk about the word linked. Have you heard this word linked before?
Yeah.
What do you think of when you hear the word linked?
They connect.
They're together. That's exactly right. They're together. That's what you got to think of. You see the word, you hear the word linked, they're together. It's not really that big a deal. When two genes are linked, that means they're together on the same chromosome. Usually, and they could, and we'll talk about that. So when they're linked, they are together on the same chromosome. All right. So, you know, A and B in this case are linked. They're together. Now, if I tell you that, let me give you some questions to see if you understand how interesting this idea is. If I tell you two genes are more closely linked than two other genes, what do you think I mean? Think about it. Think about the definition of link. Two genes are more closely linked
They can work better together
than others. What does that mean? Well, look at the definition. When two genes are linked, they're on this. When we say they're linked, they're on the same chromosome. So if I say two genes are more closely linked than others, what does that mean? They're closer on the chromosome. That's right. Excellent. Yeah. So the closer they are, the two genes are linked more closely. That means that the answer is they are closer together. Does that make sense to you? Another question is, if I tell you that there's an X-linked gene, and some of you have heard of this, haven't you? I know some of you told me, maybe not in this class, but someone said they learned it in middle school. X-linked, because you do it when you Punnett squares. The Punnett squares are this is actually a very interesting inheritance pattern. But X-linked gene, what do you think that means? They're close.
They're on the same one.
They're on what?
X.
On the X chromosome. That's it. See? This is actually very intuitive. X-linked gene is on the X chromosome. Does that make sense?
Mm-hmm.
Okay. And I'm going to take off here with these two genes that are closer linked. How can we tell if two genes are closer? How do you know?
They share similarities, maybe?
How do you know that they're closely linked? How close they are to each other? You should have a binder out. You should have your headphones off your ears. You should have your hoodies off. Like, that shouldn't even be a conversation at this point. And especially if you're walking in a little late, you have to hurry up and catch up. There's a lot to go over. How do you know that two genes are close together or not? This is actually a little harder question. This is the hardest question I've asked you all morning. How do you know two genes are close together or not on a chromosome?
Couldn't they combine or something? Too close?
I like that. What'd you say?
I said, couldn't they, combine together if they're too close to each other? They can.
That's an interesting idea. Not quite there, but it does have to do with recombination. So what do we know from meiosis? I beg you, I do not want to deal with cell phones today. I beg you not to be on your cell phone. What do we know about meiosis? When the tetrad is formed, the two copies are stuck together. They're copies of one another. One from dad and one from mom. And they're stuck together by that kinetic core there in the center. And we know that there's two genes. Let's call it, let me use a different color. Let's call it A. And let's call the second gene B. So maybe dad is big B, mom is little b, and dad is big A, and mom is little a. Remember this?
Yeah.
And they form a tetrad, right? So I'm drawing this badly
They connect each other.
because they're connected to one another. So let me connect them.
Oh, wait.
What do we know that happens in that tetrad? What is it that happens in that tetrad that increases genetic variability? When this forms a tetrad, what can happen?
They became the same thing. Wait.
It's on the board.
Their sister chromatids.
Sister chromatids. What happens to the sister chromatids?
They're twin.
Between the two homologs. This is a homolog, right? These two are homologs, meaning they have the same genes on them. The sister chromatids have exactly the same DNA, exactly the same copy, because they're copies, right? They form a tetrad in prophase 1 of meiosis, right? They form a tetrad in prophase 1 of meiosis. What happens during prophase? What's one of the things that increases the mixing up of the genes? Genetic variation, that one idea that happens. We covered it last week. Again, you must review. You see in the handout I gave you, one of my critiques, one of the things that I think you guys need to improve on is reviewing the material. You have to review it. You can't just leave.
Like 4 variations.
That's right. It increases to 4. Right now we can only if we made 4 cells out of these 4 cells, there'd only be 2 different kinds, right?
Doesn't it keep on doing it like 4 times until
Well, no. You can divide. Right. It'll make 4 cells out of these 4 chromosomes, 4 nuclei with one sister chromatid in each one. But right, if you look at it and think about it, that would only make 2 kinds of individuals, right? So, what kind of individuals would these two homologs make? 2 different. Well, pay attention to the board instead of your fingernails. Listen.
I'm not even looking at my nails.
Okay, whatever, your hands, your paper, whatever. Look on the board and pay attention. When you're looking and you see these 2 come together, if you divided each of these into a nucleus, what are the 2 kinds of beings that you would make?
A
A big A
and a lowercase a.
and lowercase a. You'd have a big A, big B, and little a, b. So let's say little a, little b. Little a is short. If you have one big B, one big A, it's tall. Okay? And let's say if it's little b, it's white. And in big B, if it has a big B, it's blue. Okay, so you say white or blue, tall or short. So pay attention on the board. Look at this. And if you divide these into 4 different individuals right now, the only thing you'd get is a tall blue and a short white. You'd get 2 short whites and 2 tall blues, right? But there'd only be 2 kinds. Are we clear on that idea? So when it forms a tetrad, what's gonna happen? How? What's the word that we use? I'm still waiting. I'm gonna wait one more second, then I'll give you the answer.
Crossing over?
Crossing over. Thank you. Ooh, that's like pulling teeth. Crossing over. But I'm glad you got there.
She got it too.
All right, back there. Good. Crossing over happens. And what crossing over happens, to remind you, is that because they're homologs and they're the same DNA, there's a break and they switch. They switch DNA. So that what you end up with is a recombined, we call it recombination, right? Recombined. That's why I said you were kind of right when you were saying the word recombine. A recombination of one of the sister chromatids two of the sister chromatids switch, and what you end up with is big A,
Has some of the little A.
and another one next to it, little a, and then this one has now the big A, and this one has the little a. That doesn't sound like a big deal until you realize that there's more than one gene on this chromosome. So now when we split these up into 4 different cells, and you should know those steps, you should remember meiosis from last week. So we have 4 cells at the end of meiosis, 4 gametes. 4 gametes, we call them gametes, which are also another word for sex cells. Remember, in biology, sex means recombining the DNA. One chromosome is going to have just the original big A, big B. So this one's going to be tall and blue. This has the genes for tall and blue. Then here you're going to have this individual is going to be tall, but it has the genes for white.
So big A, little B?
Big A, little b. And then this individual gamete, sperm or egg, is going to have a little bit of the other spouse's chromosome, and it's going to have a little a. So it's going to be short. It has the genes for short, but it has the genes for blue. And this individual is going to have the original short, genes for short and blue. Now, why do I say genes for short and blue? Because how many of these copies is it going to take to make a whole? You need 2 copies to make a whole, right? These are diploid organisms like us. We need one from mom and one from dad. This is only the sex cells from one parent. So now, this parent can now give the genes for tall and the gene for blue. This parent can give the tall and the genes for white. This parent can give the genes for short and the genes for blue. And this parent can give the genes for short and white. The genes for. The genes for. All right? Takes 2 of them. So now we got to worry about mixing these up, because now you're going to mix them up even more. You're going to recombine the DNA even more by adding another spouse, right? Another individual who's also mixed up their sex cells, right? So in any case, the point here is that crossing over increased the genetic variation.
Yes.
But let me ask you something, and I'll get to you in a second. How frequently, if I had two genes, two sets of genes, let me give you an example. And I think it helps to look at an example. If I have two sets of genes, one big A here and maybe another gene here that's B, and I had another pair of genes A, and let's make this one C. All right. So one gene A is this far away from B. One gene A is this far away from C. How often do you think there's going to be crossing over? Remember, crossing over is random.
Yeah.
Where is it going to cross over more?
A and C because it's closer to it.
You think it's going to cross over more randomly between? So you have this second.
It's A and B because they have more space.
That's right.
Yeah,
That's exactly right. It's A between A and B because there's more space in between them. If you're thinking about like these two floppy hairs floating around in a cell and just randomly flop on one another, what's the chances of the other one flopping on? Like if we had another one of these. What are the odds of this one flopping this way and crossing over or in between this space versus this one doing the same thing in between there, right?
It wouldn't be random if it was A and C because they have a more chance of
Well, it would be less of a chance.
yeah, less of
It can happen, but it's going to be rarer.
yeah.
And there's some genes that are almost next to each other, relatively speaking, that we don't see any crossing over at all between them. They say they're closely linked, and those that are far apart, we say they're linked because they're on the same chromosome, but they're not. There's a lot of crossing over. There's a lot of mixing. That's why with some genes you'll see. I'm making this up with the blue and the tall, but let's say you had a yellow, like blue and tall. There's a lot of mixing up. So you have blue and tall and blue and short and white and tall and white and short. But let's say you have yellow flowers. And every time you see a yellow flower, it's always short. Well, what does that suggest to you?
That the short one is more common.
Not really. A lot of people think that. Let's not go with dominant recessive right now. If every time you see a short one, it's always yellow, what are you thinking?
It's more
They had-
chances of getting
It was
yellow.
less likely to cross over.
That's right. Less likely to cross over, so you rarely get a tall with yellow. So yellow and tall must be closely linked. They're really close to
So
each
they're
other.
rare to get.
Whatever that genetic mutation is that makes it yellow, that must be really close to that other gene. Does that make sense? Yeah.
Like the lines, like their own.
They're genes. Yeah, the lines are the genes. Yeah, I'm sorry. They're locusts. They're locations on the chromosomes. Yeah.
You know how you said when they cross over, right? Like, they share the DNA. Like
They switch.
yeah, they switch the DNA. Is there a chance they can have like you know how you showed us how they shared one half? Can they be like more?
It could be more. It could be less. It just switches. Wherever it lands, wherever they touch,
Like if there's
they
like,
exchange because they're the same exact DNA. So as far as they're concerned, it's ATG, same thing. So they switch and they chemically switch it out. They match, so it's just not a big
so
deal
if
for
they
them.
touch, like if they like when they cross over, they touch like up here and down here. They'll both be switched to the other one. And
Yeah, each switches.
Okay.
So whatever they touch, they switch that DNA at that point.
Okay.
So what?
Do you have the boards for the deal? Can I see how big they are?
They're in the teacher workroom, not in here. But I can help you third period.
Oh, deal.
All right. Does that make sense, everybody? Anybody have a confusing point? Is this confusing? Please have her sit up. Make sure she's taking notes. So we're good with this. Now let's talk about Punnett squares. What do Punnett squares have to do with all this?
It's just basically what we just did with the
It basically lets us deal with the numbers behind the switching.
and how much percent.
And what percent chance is it of you to
that.
get that particular gene? But see, crossing over and linked genes impact that.
Because
Like if it's X linked and you're a boy, Well, it changes your percentage chance of getting that, whatever it is. If it's Y-linked, if it's a Y-linked gene and you're a girl, you have 0% chance of getting it, right?
Because females don't have.
Because you don't have a Y.
Oh yeah.
So it's not even an issue for you. Does that make sense? So where the genes are makes a difference, and how closely related they are makes a difference.
The closer they are, the rarer it is to get
The closer they are, the less random it is to get the recombination. The closer they are, the less random. But right, Punnett square is about randomness. So, and by the way, who has Mr. Pennypacker?
me.
So a lot of you, you'll be doing these random rules, these chances and percent chances, and Punnett squares in his class as well.
You said the closer they are, right?
The less random things are, like of getting the two together. The less random the chance there is of getting the two together. We call it, when you get random, how random is it for you to get one gene or the other? We call it independent assortment. It's called that's one of Mendel's laws. Independent.
Didn't Mendel test the pea plants or something?
Yes. We'll go over. I feel like going over the idea is easier than trying to introduce Mendel and his laws and trying to make it. I think it's easier to start and figure out the mechanics and then talk about the rule, rather than try to tell you about some rule that you don't have any idea about. That's my opinion. I think it's solid. Independent assortment just means it's random. Which gene you get is random. There is no, you get mom's gene or dad's gene, big A or little a, you get big A with big B or big A with little b, it's all random. Now, if they're linked, if they're strongly linked, they're close together on the chromosome, then you have less randomness.
So would it be dependent?
They're not really dependent, independent. They're just, well, I see what you're saying, because independent versus dependent. That's really good thinking. But no, it's just, all it means is that they're not, that this rule of independent assortment isn't really going to work as well with those genes that are very close together. Is that making sense? Or genes that are X-linked, for instance. That's when we look at Punnett squares, when we look at pedigrees, and we we start to expect this or that and the other thing. X-linked genes show weird things. And so now all of a sudden it's not what you expected.
Okay.
All right? So let's talk about what we expect. If they're independently assorting, what we would expect if mom is big A, little a, and dad, and let's just make it round, and then dad is little a, little a. All right, you got these two. They're going to come together, right? Well, which one are you going to get?
The big A, the dog.
We don't know, right? Because big A, that's not how the sperm comes, right? That's not how the egg comes, correct? That is, how do the eggs and sperm come? With half of that, that's right. So mom can give you a big A, or mom can give you a little a. Dad can only give you a little a. But which one? Yeah, because that's all he has. Your parents can only give you what you got, what they got. They can't give you something you don't have. They don't have. Does that make sense? Unless there's a mutation, you get what you've inherited from both parents. That's the idea of inheritance. Do we understand that? All the genes you have are a mixture of your two parents. Yes? We're good? All right. So if that was little a, his genotype, let's go ahead and add that. I think we have that word up there on the board. No, we don't. We have to add that word. Someone with good handwriting want to add it on the board? Genotype. Thank you. Genotype, Dad had the genotype of little a, little a. What does genotype mean? The type of genes. Right? So it's easy. It's very intuitive. This is all very intuitive. It's unlike all that biochemistry we did. that had all that kind of crazy stuff. We spent so much time this year. We had to spend that time so you understood this. But this is all intuitive. It makes sense. If you just think about it for a second, it really does make sense. There's a lot of kids love genetics more than some of the other topics in biology because it all just makes so much sense. So dad is little a, little a. Mom is big A, little a. Now, I say it makes sense. That's assuming you get the basics. Okay. So, but because mom and dad have this genotype, dad can make it, we're going to half it. That's the whole point of meiosis, is to half the cells, half the DNA in the cells. Dad's only going to have a little a in each sperm. Mom is either going to have big A or little a. Well, how do we know what the odds are of you getting the dominant trait? And this is dominant recessive. And let's say big A is, pick a trait. Go ahead. Pick something. Let's
Pick one.
okay, yes, but let's pick a flower because it's easier with flowers.
Oh,
Humans are so complicated.
Okay, flower.
Flower, big A is what?
Big
Tall.
Yeah.
And little a,
Short.
little a is short. Now, because it's dominant and recessive, this is really key. It doesn't matter if you have a big A, the other one doesn't matter. It's going to be tall.
Yeah, it's gonna.
For the phenotype, there's another word. This side's genotype. The other side is phenotype. What is phenotype?
The trait.
The trait,
Oh, I got it right.
which is what you see, the type of traits, right? So the trait is tall or short. If you have a big A, then it doesn't matter what the other allele is, you're going to have that trait.
Because it's dominant.
That's going to be really key for your understanding. When you start applying these Punnett squares for homework tonight, you got the sample. I'd like to try to get you doing some of it here, but I don't know if we're going to have time.
There's homework.
It was supposed to be for classwork, but we may not have time to do it. Are we clear? All right. So let's do an example. If we have mom, she can give a big A or a little a. Those are two options for mom. Dad can give a little a or a little a. It really doesn't have much of an option, but there it is. So you can make 4 different kinds of people, and plants in this case. 4 different kinds of plants. Or I don't know how many different kinds of plants. I'm sorry. There's 4 different combinations we can make. We can make big A, little a. We can make little a, little a. We can make big A, little a, and little a, little. So actually, we can only make 2 different kinds, can't we? What are the 2 kinds?
Big A, little a.
Right. So it's either 50%
Big A, little
tall and 50%
short.
short.
So it's.
So this is what we call the phenotypic ratio.
It's an equal amount?
It's 1 to 1.
Yeah,
It's 1 to 1, equal amount, 50-50.
yeah.
Percent chance of being tall versus short is 50-50 if mom is heterozygote. By the way, to remind you, mom is different, has different alleles, so we call her heterozygote. And dad has the same, so we call him what? Homozygote. Okay? So one is homozygote, the other is heterozygote. That's the phenotypic ratio. What's the genotypic ratio? What's the genotypic ratio? That's right, 50-50 or 1 to 1, 1 big A, little a to 1 little a, or 50% big A, little a, 50% little a, right? Depending on how they want you to represent the number, it could be in percentages or it can be in a ratio. Are we okay with that? And again, you'll be going over this with Mr. Pennypacker in math as well. Yes?
How come the one that has big A and little a is considered tall instead of tall?
Excellent, excellent, because that's a great question. Look at what we said the inheritance type is. Dominant and recessive. And last week we defined dominant and recessive. Dominant meant what?
Like more higher chance of having
If it's there, it
will be dominant.
it has a trait. It shows the trait. That's what dominant means. If you have that allele, you have that mutation, you show the trait. Period. No more discussion about it. That's what dominant means. To recessive means what?
Is the like it's the
You need 2 copies of that mutation in order to see that trait. So in order to be short, you need you had little a.
yeah,
It's the only way you're going to get short. These are great questions because I'm sure there's half the class had that question, they didn't ask it. Is everybody okay with that? We're good?
So I have a short trait.
So that you're different. Humans are different with our height. That's why I didn't want to use humans. Humans, our height is controlled by many different genes. So we don't show tall or short. We have everything in between. Does that make sense? It's not plants have some plants have a much simpler genetics. That's why Mendel was very lucky he chose peas. Yeah.
I don't know if you already answered this, but what if it's too dominant?
Yeah.
You want to do it? Like if we had big A versus little a. Well, dad or mom could only give one big A. This parent can only give one. That's a big A. This parent can only give
It's gonna be dominant no matter.
little a. It's going to be dominant. If there's 2 big A's, all their children are going to be dominant. Then what their genotypic ratio is depends on the other parent. If the other parent is short, so they're both recessive, then you'll get this. They're all tall, but their genotypic ratio is going to be heterozygous. Does that make sense? Huh?
What percentage would be
Say like, so I guess it would choose all the big A's, but like, say the kid that had like big A and little a, but it shows big A, like if they have kids with somebody else that has like the, I guess gene from little a,
Well, let's do it. You're actually doing it in the practice problems, by the way. So, but let's go ahead and mix it. Let's say this is called the F1 This is the parental generation, the first generation. Now let's look at the F1 generation, the first set of children. Yeah, Mendel did exactly that. He took the babies from the first generation. He took what he called true breeding tall plants and true breeding short plants. True breeding, he meant homozygous. He didn't know that. He didn't know anything about genes. He just knew every time I mix these plants with these plants, I always get tall. Every time I mix these plants with these plants, I always get short. So I wonder if I took the short and mixed it with the tall.
the short offspring?
Then I got these. I got all tall. Oh, well, what's going on? That's when he said, that means that there's something that these kids got that is dominant. It only shows up. If they have it, they shows up. Today we know that there's a mutation, but we're moving on. They said, what happens if I mix these babies? Remember, they're plants, so it's not like brother-sister, right? So what if I take the pollen big A, little a, and mix it with a big A, little a? The children of the plants
Who have dominance?
who look like they're all tall.
And they're not. There's one chance of them being right?
But, well, let's look. And these were all tall. It looked like they only, right? They're all tall. I couldn't tell the difference. But I take those children, I mix them, I combine them. Do you see that? Do you see that's what I'm doing?
But what if
All right, so then what do I get? I get
big A,
big A, big A, big A, little a, big A, little a, and little a, little a. I get 1/4,
of having
25% short, and 75% tall.
you have a 20.
This is give me a second. These are phenotypic ratios, right? Is 75 to 25. Now, genotypic is 50% big A, little a. Again, Mendel didn't do genotypic because he didn't know genes existed. Okay? All right, so big A, little a. 25% big A, and 25% little a, little a. But remember, these are all tall. That's where you get 75%. And this is the only way to get short.
So if that,
So they have a more likely chance of being tall than short.
Yes. Now we say likelihood. Like if you mix these two, there's more likely you're going to get more tall than short. And and Mendel counted hundreds. He crossed hundreds and counted them all. All right, many generations for many years. So he got these numbers to come close. The more you do it, the closer you come to the random these odds. That's how odds work. As we discussed earlier in the year, if I flip a coin, what are the odds of getting ahead? 50%. What are the odds of me getting ahead if I flip the coin? Flip the coin twice. What are the odds that I'm going to get heads twice? So it's if I'm flipping a coin a bunch of times, where the head, the first time is 50%. I'm going to put 0.5. Is that okay? 50%. The second time for me to get ahead. Well, yeah, but it's still to get heads every time I do it is by itself. If it's independent of, if they're independent, if I'm not taking a head away every time I flip it, it's 0.5. But what are the odds of me getting 3 heads in a row?
it's very
It's here's the rule. It's called the product rule. And you're going to learn it in math. And Mr. Pennypackers, I talked to him this morning about it. It's, 0.5, the odds of getting 1 by itself, times 0.5 times 0.5. So the odds of getting 3 heads in a row is 0.5 times 0.5 times 0.5. Every time you do it, the odds of it coming out the same decreases, right? Because 0.5 times 0.5 is 0.25, then 0.5 of 0.25 is 0.14 or something, right?
I don't know.
14%, half of 25%, yeah.
I'm not good at math.
Yeah, you would expect as someone keeps flipping it, if it's even, if it's fair, that eventually you're going to get a tail rather than getting 10,000 heads in a row.
And then will that like mess up the chances of getting ahead that it research?
Well, they're independent. It's just like every time you flip it, it's 50-50, it's going to be a head. But if you flip it 1,000 times, you'd expect it to get closer and closer to 50-50, so you'd expect to see some tails. Yeah. Phenotypic and genotypic. How many genes? The kinds of genes. So you have big A in this case, little a, and big A, little a. So there's 3 different kinds of combinations, right? What you see, either tall or short. Whatever the 2 options are. If it's dominant recessive, you have 2 options. In this case, in this example, tall or short. So make sure you write that down. Do those practice problems tonight. Tomorrow we'll go on to dihybrid.
Okay.
When you draw the chromosome, what's the allele and what's the gene?
What's the what?
Allele and what's the gene?
All right, so when I draw a chromosome, and this is going to be true most of the times you see it, you're going to see the chromosome. The gene is the location. That's the gene. Now, what version of the gene do I have? Do I have big A or do I have little a? Right? The version of that gene is the allele.
They're just talking about
But that gene that's it. So I just introduced him to the product rule. So we go back and look at this chart. This is, let me draw this around here. This is one egg that mom could make. This is another egg that mom can make. This is a sperm that dad can make. And this is a sperm that dad can make. So this combination, remember, all of us have 2 copies of every gene, one from mom and one from dad. This combination of genes is what came from mom and dad. That's how we read this chart. It's divided into 4 squares. So each square is worth 25%. Does that make sense? Yes? Do I have any questions? Did that explain your question, dear? Okay. So when we look at this chart, that's how we read it, and that's how we know that 75% have no disease, because these three opportunities, that are, this is the only way to get the disease, is to get two little s's. So my question is, do we have another baby? Do my wife and I do we make the logical choice to try to have another child if we have one that doesn't have disease? Are my odds increased or decreased? They stay the same. Each individual event stays the same.
So it's like each child
Has a 25% chance. Each time you roll the dice, there's a 25% chance that you're going to get two little S's. But there's the trick to this, right? There's the product rule, isn't there, ladies? There's the product rule. And what that product rule says is that if I have, every time I do an independent event, I have to multiply the odds so what are the odds of me getting a normal child, two normal children in a row? I'd be 0.75 times 0.75. Yes, but having two in a row, flipping the coin example, that's the trick. The chances are the same for each independent event, but having a bunch in a row changes the odds. So what are the odds that I'll have two normal children versus a normal child and then a child with the disease? That changes when you start to multiply those together. So genetic counseling can be somewhat tricky. All right, so you have the practice problems, they have the answers, and we'll do chi-square analysis of dihybrid crosses tomorrow. Have a good one.